Good evening, everyone. My name is Savannah Mapes, and I will be your host this evening. I want to welcome you all to our virtual Scientist Walks Into a Bar event. Tonight, you are going to hear from 3 graduate students from the Virginia Institute of Marine Science, or as we call it, VIMS. For those of you who are not familiar with VIMS, it is a research institution located in Gloucester Point, Virginia, that has been focused on coastal and estuarine science in the Chesapeake Bay and beyond since 1940. It is also the Graduate School of Marine Science for William and Mary. VIMS has a 3-part mission: to conduct research, educate students and the public, and provide advisory service to the state of Virginia. We offer many public education programs throughout the year, such as tonight's event, and we would love for you to join us for more of our programs. You can find out about all of our upcoming events and programs in our website at www.vims.edu or by signing up for our monthly e-newsletter, e-tidings. Scientist Walks Into a Bar Graduate Student Edition started as a VIMS graduate student version of an existing event that has gained popularity in recent years. We've previously held these events in local breweries and tap rooms, but as you can imagine, that's not really possible at the moment. But the show must go on, so we have adapted our event to bring it to you virtually tonight. For tonight's event, we will have 3 graduate students who are technically still first years going into their second year this fall at VIMS present to you, each providing a 5-minute fast talk about their upcoming research, followed by 10 minutes for questions and discussion. Before we get started, we'd like to thank our event sponsors: First Advantage Federal Credit Union, Chesapeake Bank, Hogg Funeral Home, Langley Federal Credit Union, Rick and Libby's, and Whitley's Peanut Factory. Thank you so much, and we truly appreciate your support. Now, without further ado, let's get to our program. Our first speaker of the night is my good friend, Abigail Sisti, a graduate student in the Biological Sciences Department at VIMS. Abby received her bachelor's and master's degrees from the University of Alabama. If she could be any marine animal, she would be a decorator crab because she too likes to show off all of her thrift store finds. And with that, let's welcome Abby to the screen.
Hi, everyone. Thank you for tuning in today. Again, my name is Abigail Sisti, and I am a first-year PhD student here at VIMS and a recipient of the Harry and Barbara Hager Fellowship. And for the next few minutes, I'm going to be sharing a little bit about one of my study organisms, the American lobster. You may better know them as the Maine lobster, but I like to think of them as one of the species showing us the most immediate effects of climate change. In addition to being a cultural icon, the American lobster also plays an important economic role in New England. In this graph of American lobster landings, we have year on the x-axis and millions of pounds on the y-axis. And you can see that lobster fishing has increased dramatically since about the mid-90s. In fact, in 2018 alone, the value of the lobster fishery was over $400 million just in the state of Maine, representing over 70% of the commercial landings in that state. But lobster populations aren't just changing in the Gulf of Maine, they're changing throughout the lobster's range. In this animation, you can see lobster populations with areas of high density shown in purple from 1967 to 2014. And one thing is clear, lobsters are shifting north. But this isn't just happening at the furthest reaches of the lobster's range, but as far north as southern New England. In these graphs, we have southern New England on the left and the Gulf of Maine on the right. And the abundance is shown over a period of years from 1979 to 2018, with the abundance of lobsters in millions on the y-axis. From this graph, we can see that while lobster populations have been increasing dramatically in Maine, they've also been declining dramatically in southern New England. So why is this? Well, to put it simply, temperature. Lobsters like it cold, and with climate change, warming temperatures have shifted the hospitable conditions for lobsters north towards the Gulf of Maine. But is this really good news for Maine? Well, the Gulf of Maine is changing quickly. In this figure, we see global sea surface trends over the period from 2004 to 2013, with this bright red spot showing warming as the Gulf of Maine. In fact, the Gulf of Maine is warming faster than 99% of the rest of the ocean. And that's not all. Climate change isn't just about warming, but also about ocean acidification. or the process by which CO2 from the atmosphere is absorbed into the ocean, where it goes through a series of reactions that result in slightly more acidic water. Now, the Gulf of Maine has recently been described as being more vulnerable to ocean acidification due to the cold, fresh waters that enter the region from Nova Scotia, and these waters are more likely to absorb that CO2 from the atmosphere. So one thing is clear. As we move forward into the future, lobsters will have to deal with two major stressors: elevated temperatures and ocean acidification. We don't know much about how lobsters will respond to ocean acidification, but there is evidence to suggest that different life stages will be affected differently. And one area that's been very understudied is how ovigerous or egg-bearing females, like the one shown on the left, will respond. Now, these females are important because they form the basis of an important management strategy for protecting the lobster fishery in the Gulf of Maine. So this is the area that I'm interested in focusing on for my dissertation. I'll be answering questions like, under these climate change conditions, how will those ovigerous females find food, keep their eggs healthy by grooming, and how will those eggs themselves grow and develop? The answers to these questions will not only move forward how we understand the impacts of climate change on an important commercial and cultural species, but also help inform management decisions that will ensure the long-term viability of the fishery in the future. And with that, I will take any questions.
Thank you, Abby. So I will be facilitating any question answering. How many eggs does a lobster have?
Oh, lobsters can hold thousands of eggs in their egg brood at a time, up to 10,000. Yep.
We have another question from Venus Spencer. Are you from Maine? Interesting subject, and why did you choose this topic?
Oh, that's a great question. I'm not originally from Maine. I do have lots of family in the New England area, and I have lived there before when I was very young. But I saw an interesting opportunity to explore a species that we will have the opportunity to bring into our lab and kind of expand my impacts in research beyond just the Chesapeake Bay, although that's something I'm also interested in.
We have another question from Lorena Clark. I do apologize if I pronounced that wrong. But do you have any idea why the Gulf of Maine is getting warmer than the rest of the areas around it?
Oh, that's a good question. So I would first say that it's not necessarily all areas are warming, and the Gulf of Maine was colder to begin with. So the effect is different for lobsters in the Gulf of Maine versus another region. But why specifically the Gulf of Maine is getting warmer is at least partially due to the current structure in the area and different physical processes that I know much less about.
Another question from Lisa Muras. Are lobsters making any adaptations to the warmer waters?
This is a good question. So one of the issues with warm waters when it comes to lobsters is at about 20 degrees Celsius or approximately 68 degrees Fahrenheit. They deal with respiratory and immune issues as a result of those warming temperatures, and it also makes them more susceptible to disease. So there is a limit on their ability to respond to those warming temperatures, but it's something that definitely needs to be investigated further because, like I discussed in my talk, lobsters can be found far further south than New England. So it's possible that different populations are better adapted to warmer temperatures when they've already lived in them compared to those that start in colder temperatures, like in New England.
We have another question. How long do you think your research period will last?
Well, lobsters can, the period of time in which those ovigerous females are bearing their eggs can last from 9 months to 11 months, depending on the temperature, and could be even shorter if we're exposing them to extreme elevated temperatures. So because that's the life stage that I'm studying, I plan to expose lobsters for that approximately 9 to 11 month period.
And we have another question from Brian Kim. Do you think lobstermen in Maine should start looking at alternative fisheries or start looking at aquaculture?
So some lobster fishermen do set their traps to collect other species in addition to lobsters, and reports I've seen have shown that diversifying those fisheries are important to some lobster fishermen. In Maine specifically, conservation strategies have a really great history. So lobster fishermen in the Gulf of Maine seem especially receptive to different conservation strategies. So that could definitely be a direction in the future.
Yay, I get time to ask my question. What experiments do you want to do to answer your research questions?
That is a great question. So our lab is receiving lobsters from the Gulf of Maine, and we're hoping to expose them to different combinations of ocean acidification and warming conditions. And for my research, I plan on examining the behavior of those ovigerous lobsters, as well as the physiology of their eggs. So their survival and the different processes they have to deal with stressful conditions.
Great. We have another question from Melina. How many species of lobster are there along the Atlantic coast? And if there are several, do their life histories differ significantly between latitudes?
So I'll specifically be focusing on the American lobster, and its range goes from Nova Scotia all the way down to North Carolina, but there definitely are other species of lobsters. There's the Caribbean spiny lobster, which prefers lower latitudes. There is the European lobster, and a few others. So all of those lobsters do have slightly different life histories and are affected differently by the temperature, just because they are different species who live in different climate conditions. So there's been increasing studies about how climate change will affect them, but it seems similarly that so far evidence suggests that ocean acidification and climate change can increase metabolic rate. So similar, if you're outside in warm temperatures, your heart starts beating faster, you might be breathing a little bit more heavily because of those increasing temperatures. But there are definitely species-specific responses as well.
Okay. We have a question from my mom, Sheila King. How do you anticipate the warmer temperature to affect the food lobster sources and the developing young?
Yeah, this is a great question. So for the developing young lobsters specifically, there has been some warming temperatures are expected to increase development time, whereas there's been some evidence that ocean acidification can actually slow down or reduce development. So we're still exploring how those two things interact to affect the lobster juveniles and larvae. In terms of their food sources, that's an important consideration moving forward, because the food sources available to a lobster, such as zooplankton, may have different timing in their availability. So it's important to consider when those eggs hatch versus when their food is available for those young lobsters. And if there's a mismatch because of climate change, then that could definitely impact the number of lobsters that are able to survive and contribute to the population.
We have a question from Cynthia Edwards. How many out of the 1,000 lobster eggs will survive to become adults?
So there are more than 1,000 lobster eggs. There's usually several thousand on a lobster brood, but only a very small percent. I don't know the exact number, but only a very small percent of those will survive, which is one of the reasons they have so many. If you put out as many larvae as possible, then the chances are good that at least a couple of them will survive.
We have another question from Lorena Clark. I will quote this question. This will sound strange, but do you just call the fisheries in Maine and ask them to just send you lobsters for research?
Oh, that's a great question. So there is a permitting process that goes through the Maine government, I believe the Marine Department of Resources in Maine. So there is a limit on the number of lobsters that we can ask for to study. And once we request that number of lobsters, we do have to pay for them and they are delivered to us.
Okay, and we have yet another question from Brian Kim. Is there a northward migration of tropical lobster species similar to northward migration of North Atlantic lobsters?
This is a good question. So first, I would say I wouldn't call it a migration per se. It's more that the lobsters in the areas in the south are declining because conditions aren't as good for those lobsters to grow up and enter the population, whereas in the north, the population is growing for the opposite reason. Conditions are better, so more of those young lobsters can grow up and join the population. So we see a shift in the size of the population so that it is geared more towards the north. So in terms of, it's not quite a migration, but we are seeing in species, aquatic and terrestrial, across the globe, shifts in those population numbers similarly to move northward as, or not just northward, depending on which part of the world you're in, towards the poles in response to warming temperatures.
All right, thank you so much, Abby. You're obviously very knowledgeable about this and have some really cool stuff to come. So hopefully you'll give another one of these talks with some research findings very soon.
Looking forward to it.
All right. Thank you so much, Abby. That was such a fun presentation. On to our next speaker, another good friend of mine, Alexandra Schneider, or Alex. She is a graduate student in the fisheries department here at VIMS. She received her bachelor's degree from Villanova University. And if she could be any marine animal, she would be an ocean sunfish, because she would just love to bask in the sun all day. Me too. Now let's get crabby. Alex, the screen is yours.
Thank you, Savannah, for that lovely introduction, and for everyone for virtually joining me this evening for my talk on blue crab reproduction in a changing Chesapeake Bay. Understanding biological shifts due to climate change and warmer temperatures is really important so that species can maintain the roles they play in our society. The blue crab has many important roles in the Chesapeake Bay region and the east coast of the United States. Economically, the fishery has a $219 million yearly dockside value. Ecologically, it is a common predator and prey to other economically and ecologically important species throughout its various life stages, and it also plays a really important role in the culture of the region. So let's get right into reproduction. Female blue crabs can only mate once, and mating typically occurs in the inner tributaries of the bay where the salinity is lower. Mating peaks in April and May, after which females migrate out of the tributaries to the spawning grounds in the lower bay from April through November. The spawning grounds really just have a higher salinity, which is more conducive to egg and larval crab development, and once in the spawning grounds, crabs will remain there and continuously produce egg masses or broods for the entire spawning season, about April through November. And it's thought that crabs in the Chesapeake Bay will have 1 to 3 broods per season. A female with an egg mass is colloquially called a sponge crab because, as you might notice, the egg mass, which is right here, kind of looks like a sponge. It takes 10 to 15 days for a sponge to fully develop from a bright, vibrant orange to a brown to a dark black shade, which the color transition is a result of the larval crabs developing and consuming that yolk. And if we take a look under a microscope and look at the eggs developing, we can see those larvae. Those big black dots are their eyes, and the little black specks are their spine. These animals have a ton of offsprings in a single egg mass. There can be anywhere from 0.5 to 3.5 million eggs per sponge. And once those eggs hatch, larvae crabs are the results. They're called zoea. They don't exactly look like crabs as we would think of them. They're a little bit closer to little aliens, but I think they're really cute. They have those long spines that are meant to protect them from predators, and long tails and swimming arms used to orient themselves in the water column. And it takes about 40 days for these zoea to develop into a more crab-like form when they metamorphose. And really all this background just boils down to timing. Reproduction is all about timing. When a female first mates, when the crabs migrate, when she first spawns an egg mass, and how long it takes for those eggs to develop and for the zoea to develop are all about timing. And this timing is heavily dependent on temperature cues. The effect of temperature can be seen if we look at the blue crab's range, which in the United States spans almost the entire East Coast. This also encompasses a wide temperature range, and we can see that northern crabs have a longer winter and shorter growing and reproductive seasons, while southern crabs have a shorter or no winter season and longer growing and reproductive seasons. Along this latitudinal temperature gradient, there are differences in blue crab reproduction. For instance, in the Chesapeake Bay, females have 1 to 3 broods per year, larger eggs, and fewer eggs per brood. Crabs in North Carolina have 5 to 6 broods, and crabs in Florida have 7 to 9 broods, smaller eggs, and more eggs per brood. There are a lot of other factors that go into the differences along this range, but I'm running an experiment that looks specifically at the effects of temperature. So I'm holding 32 crabs at 2 different treatments. I have a control treatment, which is represented here in blue, which experiences temperatures that mimic the natural temperature changes of the Chesapeake Bay season, and a heated treatment, which is represented in red, that is maintained at 3 degrees Celsius higher than that of our control treatment, and is the projected temperature for Chesapeake Bay by 2050. I'm holding these crabs to document the effect of temperature on reproduction, and I am anticipating a similar shift that mimics that of the southern crabs in the southern extent of the range, with my heated treatment having a quicker onset to spawning, smaller eggs, more broods, and more eggs per brood compared to an expected later onset of spawning, smaller eggs, fewer broods, and less eggs per brood in my control treatment. As temperatures continue to warm, the growing and spawning seasons of blue crabs in the Chesapeake Bay are expected to lengthen and become more similar to those in the southern extent of their range. To maintain effective management strategies and preserve the economic, ecological, and cultural roles of the blue crab in a warmer Chesapeake Bay future, we must understand potential shifts in biological timing, and my research is aimed to look at those reproductive shifts. So if anyone has any questions, I would be happy to try to answer them.
Thank you, Alex. I just love those little aliens. They're so cute.
I could stare at them under the microscope all day.
We already have our first question from Lorena Clark. Is there a difference in the survival rate from the north cooler waters and the southern warmer waters?
There isn't a necessarily difference in survival rate in terms of adult crabs or larval crabs. A lot of factors that affect survival for crabs is disease, fishing pressure, predators, and that changes throughout different areas that they can be in. It's not necessarily a temperature-driven process. It is more just a habitat-driven process.
Okay. We have a question from Jeff Shields. Do you eat crabs? And if so, how do you like to prepare it?
I have to say I do not eat crabs. I think I've dissected a few too many to ever find them appetizing again. And honestly, I'm a little offended that one of the giveaway gifts is a crab mallet.
We have a question from Evan. What made you want to study blue crabs?
I started studying blue crabs through an REU I did a couple years ago. Out of Rutgers University, and I just think that their life history and the complex migration and larval development offshore is really fascinating, and they are quite entertaining animals, so they stole my heart, I guess.
They are quite cute.
Something.
We have a question from Venus Spencer. Are bigger crabs better, or do people just prefer bigger crabs?
I think that when people eat crabs, they definitely prefer bigger crabs, more meat and all of that. In terms of reproduction, bigger crabs actually can have a higher fecundity or produce more offsprings. So it can be beneficial to have larger females in the population because they'll be able to have larger broods. But other than that, I don't know anything about the size of the crab and the taste of the crab.
Challen has asked a question. Blue crabs are notorious for being problematic experiment subjects. Do you have any good escape stories or interesting experiences during this experiment so far with these animals?
Yes. They are quite escape artists. I feel like Challen might have a couple stories that he's referring to. There was recently a 4th of July Day coup where the crabs also wanted their freedom, and a few of them escaped their tanks. They're quite great at using their claws to climb out of things. So a lot of experimental troubleshooting is how to keep crabs in their tank. So be aware if you ever want to experiment with crabs.
Lisa Muras has asked, what, if any, is the advantage to having smaller eggs versus larger eggs in a brood?
There isn't necessarily an advantage. It's likely that larger eggs would be more advantageous for the larvae themselves because they can have more yolk, which is a higher lipid content, so the larvae have a little bit more nutritional quality. However, there's something to be said about many small eggs versus a few large eggs, and whether or not that individual female is putting more of her reproductive energy into more eggs that might have a lower nutritional quality or vice versa. So there's no yes or no answer to that, but there's definitely pros and cons to both.
Okay. Casey Duplantier has asked, how many seasons will a crab live?
So it's likely that these females don't survive past one reproductive season. They might get 1 or 2 or 1.5 reproductive seasons in. By the time they're actively spawning throughout a season, they're a little bit on the older side, and there tends to be a pretty high fishing pressure for adult crabs in the bay, so not many of them survive past 1 or 2 spawning seasons.
Someone has asked for a quick clarification. What is an REU?
Oh, I'm sorry. It's a research experience for undergraduates. It's a kind of like an internship through the National Science Foundation where undergraduates can do research projects. And VIMS has a great REU program that I would highly recommend to any undergraduate.
Brian Kim has asked, is the difference in brood quantity and size purely driven by environmental differences? In other words, if you placed a brood crab from farther south into a northern environment, would the brood quantity and size change accordingly?
I think it's based on the environment, not the crab itself. One of the reasons that Florida crabs can produce 7 to 9 broods in a single season is because their season is significantly longer than the season is here. As I mentioned before, with that latitudinal gradient, crabs in the southern extent of their range have a much longer reproductive season, so it's likely they just have more time in conducive temperatures to continuously produce broods. And they don't have to worry about surviving the winter, which can be a significant cause of mortality, those cold winter temperatures.
Okay, Melina has asked, how does blue crab diet differ between northern and southern populations? And how does the diet or nutrient composition affect spawning?
I don't know too much about how diet affects spawning, other than the fact that I'm feeding my crabs what I like to think of as a well-rounded diet. They're technically opportunistic omnivores because they will eat anything they can get their claws on. So I don't know of any latitudinal differences in diet. They're scavengers. They'll eat bivalves, small finfish, detritus, plant matter. So my crabs get a healthy diet of oysters, bay anchovies, squid, and seagrass.
So Aman has asked, do you think increasing the temp will cause them to metamorphose sooner than they otherwise would, i.e. lead to a premature crab as a shorter than usual development time will have other implications, such as disease susceptibility?
I don't believe so. The time that zoea take offshore, it's typically offshore development. They don't develop within riverine systems or higher up in the estuary, so the temperature is a bit more stable than compared to directly in the estuary, and their metamorphosis to their second larval stage and juvenile crab stage is mostly driven by finding suitable habitat.
Okay, Aman also wanted to know what the control temperature is for your experiments.
So they're actually in a flow-through system. So the crabs themselves are isolated in tanks because they are quite aggressive and we want to keep them away from one another. But the tanks are sitting in a water bath that has a flow-through water system. So whatever temperature the bay is right now is the control treatment, and then the heated treatment is 3 degrees above that.
One more question, Sartak Kumar. Again, I apologize if I pronounce that incorrectly. How does low salinity favor reproduction conditions for blue crabs?
So it's a higher salinity that is more conducive to reproduction. The females will migrate out of lower salinity zones to higher salinity zones. When the eggs hatch, those zoea go offshore to high salinities to develop. So the females are just bringing their eggs a little bit closer to that.
Okay, and I think we can fit one more quick question in here. Meredith Seeley has asked about the geographic differences, and are there any correlations between number of broods and egg size and population size in different areas?
So it's thought that the more broods a female has, there'll be a decreased egg viability as she continues to produce egg masses. There is an idea that this could be due to sperm limitation. These females can only mate once, so they do have a limit on how many eggs that they can produce in their lifetime. So you do see a decrease in the size of the brood and the viability of the egg as each successive brood is oviposited. Yeah.
Okay, I know there are still questions, but we must move on. I'm sure she would be happy to answer any questions you have by email or potentially face-to-face conversation. I know everyone is looking forward to that kind of stuff. But again, really looking forward to the results of your experiments and hoping you'll come back to talk to the public again about that.
Thank you.
Awesome presentation, Alex. Thank you so much. I think I've learned so much about crustaceans in the past 30 minutes, so much so that more than any other amount than the rest of my life. Anywho, what I'm trying to say is great job, Alex and Abby. Our next and third final speaker is another good friend of mine, Rachel Dixon, a graduate student in the fisheries department here at VIMS. Rachel earned her bachelor's degree at the University of North Carolina, Wilmington, and her master's degree at the University of Delaware. If she could be any marine animal, she would be a flying fish, which is my favorite fish. So she could breathe underwater and leap through the air. How cool is that? Well, welcome, Rachel. The screen is yours.
Alrighty. Thanks, Savannah. Hi, everyone. My name is Rachel Dixon. I am a PhD student in the Fisheries Department at the Virginia Institute of Marine Science. And today I'll be sharing a portion of my dissertation research with you where I am exploring the link between a species' productivity and the areas that they inhabit early in life. My research focuses on one of the most iconic fish species in Chesapeake Bay, striped bass, also known as rockfish. So why study striped bass? As one of the top predators in the bay's marine food webs, they are ecologically important, but perhaps they're better recognized as supporting lucrative recreational and commercial fisheries, both in the Chesapeake Bay and along the Atlantic coast. Back in the 1980s, the population actually collapsed, but even though it has been recovering since the mid-90s, there might be new causes for concern. In recent years, the numbers of adult fish in the stock have been decreasing. While we don't yet know what's causing this, one reason might be due to a change in the extent of high-quality habitat areas for juveniles. Healthy habitats are important drivers in the numbers of young fish produced each year, especially in the wetlands, rivers, and streams that serve as nursery spawning and forage areas. The Chesapeake Bay provides spawning and nursery sites for several important species of fish, including striped bass. In fact, for the Atlantic stock, up to 90% of the nurseries for these fish are located within our bay. Striped bass have a pretty unique life cycle. It's a term we call anadromous, which means that they spend their adult lives in saltwater but migrate into freshwater to spawn, a lot like salmon. Every spring, adults come into the bay's tidal freshwater tributaries to spawn around April to June. And after hatching, the juvenile fish then spend the first year of life in nearshore estuaries where they undergo a period of rapid growth and development. These nursery habitats provide many important functions. They need to have optimal environmental conditions, including temperature and oxygen. And to have ample food resources for juveniles in their first year, this includes zooplankton and small invertebrates. And it needs to provide refuge from predators like larger fish and birds. However, not all of these habitats are created equal, and their availability and quality can change over time. Fish habitat is strongly influenced by a broad range of natural and human factors. Annual and seasonal environmental fluctuations can lead to changes in fish distributions and affect the availability of good habitats. As an example, you can see here on the left, in deeper waters, high temperatures near the surface and low dissolved oxygen levels, or hypoxia at depth, can lead to what's called a habitat squeeze. Urbanization and shoreline development can also lead to habitat loss by greatly reducing or removing critical habitats like underwater grasses. This can also reduce water quality through sedimentation, erosion, and runoff from agricultural areas or impervious surfaces in urban areas. So when we look to improve our understanding of how these factors affect the production of juvenile fish and how many grow up to reach the adult population, we need to consider if striped bass have access to both an adequate quantity of nursery habitats and whether these habitats are of a high quality. So for my research, I'm going to be using information from a few different sources to identify and map habitat. Each year, researchers at institutions like VIMS conduct surveys to monitor the relative abundances of juvenile striped bass at particular sites in nursery areas around the bay. So these surveys tell us where fish are being found and in what numbers. I can then couple this field data with a series of modeling tools that tell us the environmental conditions when and where these fish are captured during their first summer of life. But since we're not able to find and catch every single juvenile striper in the bay, we can then instead create what's called a habitat suitability model. This model can then be expanded to the entire bay to visualize and map the habitat conditions that promote the survival and production of juveniles, and importantly, tell us which areas are consistently providing those high quality habitats through time. So 3 takeaway messages that I'd like to leave you with. First being that nursery habitats support the survival and growth of young fishes. These habitats are really important because they help protect these fish and increase the chances that they will grow up and become part of the fishable population. Several environmental factors can impact habitat condition, changing the quality and quantity of suitable habitats over time. And identifying habitats important to striped bass during these critical early life stages may help us understand how the population will respond to future environmental fluctuations, and we can then work to conserve and restore these prime locations. So with that, I'll close by sharing that if you'd like to learn more about the diversity of fishes in the Chesapeake Bay and the different fish surveys that VIMS uses to monitor and study fishes and their habitats, I'll be giving another talk through Zoom this upcoming Tuesday at 2:00 p.m. with the Williamsburg Regional Library. This talk is free and open to the public, so if you're interested in seeing some really cool fish pictures, I encourage you to join. So with that, thanks very much, and I'd be happy to answer any questions.
Thank you so much, Rachel. I just got to say, modeling just blows my mind.
I have a lot to learn, so it's new to me as well.
I'm looking forward to it. So we already have our first question from Monica Stegman. What can cause hypoxia that is not a desirable habitat for striped bass?
A good question. So hypoxia is the term given to any area of low oxygen, and usually it's associated with the overgrowth of certain species of algae. So when these algae die, sink to the bottom, and decompose, it depletes oxygen in the water column. But the problem that comes with hypoxia is that when temperatures are warmer or when there's a lot of nutrient input, it really stimulates this algal growth. So it can lead to a higher frequency of hypoxic conditions. And some fish are a little bit more tolerant, but others like striped bass are not so much. So it can change the habitats that these fish can occupy and really reduce the space available to them.
Okay, we have a question from Venus Spencer. You mentioned that there was a decrease of fishes in the 80s. What happened in the 80s that caused the decrease?
So the population decreased over time, mainly due to overfishing, and the numbers of the adult fish got so low that there's actually a moratorium put in place in the 1980s to try to help rebuild the population. And in 1995, the numbers of adult fish had reached a high enough level that the population was considered restored. But today, the adult stock is back to being declared overfished with overfishing occurring. So we're not quite sure what's going on, but there are likely a number of factors involved.
We have a question from Kai Feliciano. Are striped bass only found in the Chesapeake Bay?
Good question. They are not. So striped bass are found all along the East Coast from the St. Lawrence estuary up in Canada all the way down to Florida, but they are most common from Maine to North Carolina. So that adult stock that I just said was overfished is actually contributed to from many different populations from the Chesapeake Bay, as well as the Hudson River, Delaware Bay, and Roanoke River. But the Chesapeake Bay is the largest contributor. So the bay is a great place to study striped bass because what is happening to the stripers in the bay can tell us a lot about the overall population.
Becky asks, what are the best environmental conditions for striped bass?
Another good question. So there's 2 points in a young striped bass life where environmental conditions can come into place. So when they're eggs and larvae, the environmental conditions that have the greatest influence are temperature and freshwater flow. So usually we see higher numbers of striped bass during wet, cool springs. So those are the biggest physical processes, but then as the fish get a little bit older, the conditions that affect how many juveniles grow into adults, the temperature and freshwater play a big role, but you also have other biological conditions like how much prey is available.
Sartak Kumar has asked, what exactly is the squeeze zone?
So the squeeze zone implies that if you have hypoxia along the bottom, so an area of low oxygen, the fish can't really inhabit that area. But if you have really high temperatures near the surface during the peak of summer, or as we look ahead into the future due to climate change, that water is really too hot for those fish. So the area in the water column that the fish can inhabit gets squeezed.
Okay, Lorena Clark has asked, are you finding major differences in prime habitats between the northern and southern areas of the bay?
Good question. To be determined. This work is in early stages, but that will be really interesting to see.
Ram has asked, where's the best fishing spot for striped bass in the York River?
Great question. And although I've spent a lot of time with trawls and seines, I'm not a big fisherwoman, so unfortunately that's insider knowledge I don't have.
Kathy Boomer has asked, can you talk about the spatial resolution of your model and its potential for informing shoreline management?
Yeah, great question. So the model that we're using is developed by another group, a model of the bay. So the spatial resolution, I believe, is a 0.5 meter vertical resolution. Yes, I believe off the top of my head, that's what I know. But in terms of shoreline development, we will be able to get into the shallows to see what kind of impacts that have, but that is a good management relevant question that hopefully we'll be able to answer with work like this.
Taylor Walker has asked, can you talk about this transition striped bass make when moving from saltwater to freshwater? And do they undergo any morphological changes during this period?
Yeah, so when they're moving from saltwater to freshwater to spawn, the adults will migrate up into the tributaries of the bay, the freshwater tributaries. They do not undergo any morphological changes. Yeah, that's a good question.
It's either Gary or Jerry, and I'm sorry if I got it wrong, but asked, how long does it take for fish to reach adult stage, and how long do they live?
Striped bass can live anywhere from 10 to as long as 30 years. Usually the juveniles will spend the first 2 to 3 years in these habitats before joining the adult population and starting to undergo that spawning migration.
Kay Knotts has asked, besides your graduate program, who else do you see yourself sharing your findings with?
Well, this work in particular is being done in collaboration with a lot of different management agencies across the Bay, including the Chesapeake Bay program. So it's particularly exciting. We'll be able to take the results of this research and get it right into the hands of the folks that are implementing these management decisions for this species. So it will be good to see what comes out of that.
And one last question. What can East Coast citizens do to help striped bass juveniles?
Another great question. So I would say the two biggest things that people can do to help striped bass, number one, make sure you're following your proper fishing regulations. So that includes size limits, seasons, gear restrictions for wherever you live. And second would be to work to prevent pollution from entering your local waterways and impacting that habitat quality, especially those nearshore habitats that support fish nurseries. So things like installing green roofs or rain gardens to catch rainfall, you can use porous surfaces like gravel in place of asphalt or concrete. So things like that, and protecting our nearshore habitats for these fish.
Okay, thank you so much, Rachel. I am so looking forward to seeing how your research comes out and that model as well, which I'm sure is going to blow my mind as well. But you too are definitely welcome back to share your research with everyone here.
Thanks.